is. So my talk is going to be a little bit different today. It's going to be very kind of focused in a specific area, obviously CRISPR. And I'm essentially in a commercial extension of Cardea, meaning we have an exclusive license to use the exact technology in a singular way, but all the functionality, all the principles that Francie elegantly went through apply to our work here at CRISPR QC. And I'll redundantly show a couple things in a slightly different way, which is probably not a bad idea, but this whole talk will be focused around our technology or our capability, which is really designed to empower CRISPR researchers to design and implement checkpoints that will allow for better outcomes related to editing. So this slide is nice in the sense that it really helps me convey to the audience that where do we sit in the CRISPR workflow? Because the CRISPR workflow, if you are a CRISPR researcher, is quite complicated. And it really kind of has a breakpoint, I see, where you would, oops, oh, that's not good, Ken. Where you would typically focus on a in silico portion. So as Francie mentioned earlier, when she briefly stopped on the CRISPR area, she talked about guide RNAs. Well, guide RNAs are essential in the process of developing a final goal of editing a cell line. And they are designed by inputting sequences or loci information into in silico software. And actually what that does is it kicks out several different recommendations of guide RNAs that could be used for this editing event in cells. Now, why do we get multiple options of that? Well, one way to interpret that is that it may not be an entirely exact science that's predictable to the best guide RNA design to maybe produce the best editing outcome. And that is partly what I'll show you in a minute when I kind of go through the different types of assays that we do and how we extract useful information out of there that becomes valuable to the CRISPR researcher. So kind of to the left where it says project submission and guide design and guide synthesis, that's where I was really talking about. Now, where do we fit in? Because after the guides are synthesized, you really go through an arduous process and painstakingly committed set of events to get down into cells. And really until you complete the entire process, it's not really easy to predict whether you're on the right path or not. So what we do at CRISPR QC is we operate in this box that you see on this slide, which really is separating the pre-cellular work from before I go into cells. And the things that we do that may be unique and not always easy to obtain in the CRISPR field is we will look at things like enzyme quality control. What does that mean? That actually means we're just looking at Cas9, Cas13, Cas12, whatever it is. And like a lot of proteomic work, since CRISPR is unique in that you've got half of it's a protein, an enzyme, the other half's DNA or RNA, you really have to always think about, is my protein portion, my CRISPR, impacted by my Cas9 or the Cas enzyme of interest. So we do things like look at reproducibility, lot-to-lot variation, storage, formulation. All these things are capabilities that are possible with CRISPR QC. And I'll show you one example of that in a minute right after that. We also take that a step further and look at guide RNA performance, and that is based on this guide RNA synthesis that occurs, and you may get 7 to 10 different guides to make a commitment. Am I going to do all 12 moving forward? I'm going to do 4 or 5. How do I make that decision? We even take it a step further, and I'll show you data as well with the guide RNA, but I'll also show you data with the CRISPR cleave capability we have, which means we can actually mimic a cleavage assay of an amplicon that represents a loci within, a stretch of genome that you're interested in. And then more recently, we've been able to start diversifying our capability to look at pH and how pH may affect a CRISPR molecule or an RMP, and also multiplex capability. So is everyone hearing me okay? I got a little background. I don't know if that's coming from my end. I'm in a room, but is everything okay on the sound and everything?
I hear you well, Ken.
Okay, I just wanted to make sure it wasn't coming from me. Okay, I'm going to move to the next slide. Francie had a slide exactly like this, and again, I just want to emphasize that Kiara Aren is actually our CSO here at CRISPR QC, and it was this 2019 paper which really kicked off the excitement that perhaps a CRISPR chip type technology really could be generated, optimized, and commercialized to empower CRISPR researchers to learn a lot more about their assay designs in standard checkpoints. We're all about creating standards of reproducible capability with our technology for our CRISPR researchers. Francie showed this as well, and it's a great slide. And I'm not going to go into it because I think you're probably familiar with it. We use the exact same BPU that Francie showed, an array of possibilities and applications to use it for. We just focus and stay, in that vertical of CRISPR. So I don't get to venture into a lot of different sandboxes. I'm kind of only in the CRISPR sandbox, and that's what this whole talk will focus around. So we actually, if you see, the only difference in this slide is down in here, there's actually a cartoon of a CRISPR molecule, and that's what always ends up being on our chips, and that's what we always assess. We're either assessing Cas, guide RNAs, amplicons, cleavage, all of that, or a part of that. And I'm going to show you actually how we actually make one of those chips, because I think that question came up. I wrote a note down and someone asked, how do you actually functionalize? And I think Francie answered that really nicely in that I think someone said there's pixie, maybe some pixie dust or magic, and there actually isn't. As much as I would like to say there is, it's pretty straightforward, as Francie explained, for functionalizing a ligand of interest. And I'm going to take you through how we create a CRISPR chip. And this is the only chip I ever create. Every day I come to work, this is the chip I'm creating, not going beyond this. So, let me back it up. So I'm only on one transistor out of that array of 3 channels and those 5 times 3, 15 redundant ones. But you do see here the graphene design that Francie explained very nicely. So these are what the chips come to us in the lab. What we do is we take the pyrene. So the magenta with the yellow is actually a pyrene, but in this case, it's got a butyric acid or a carboxyl group. And the nice thing is to, get backtrack to Francie a little bit. These pyrenes are actually commercially available, and they do come in a large range of conjugation capabilities. So we use pyrene butyric acid almost exclusively. And what we do is some very old standard biochemistry cookbook stuff. We activate with EDCNHS. That creates an active ester at the end of that yellow carbon chain. We then bring in Cas9 and we go through a standard amide bond. So that's going to become a covalent interaction. You may not see it up here, but we do need to block any unused ones. Chemistries are always not 100%. We block it typically with your standard amines or perhaps a BSA protein. And this right here, to be honest with you, is a Cas study in some regards, because if we bring in a guide RNA, and hopefully that you saw that float in, that's the first time that we see a direct interaction between the Cas molecule, which has been tethered to the chip, and a guide RNA inserting or binding or interacting with a Cas molecule. Now, we can certainly take this further and we can bring an amplicon in. And after that, if we would like to see the amplicon binding, we can measure that. And then subsequent to the binding, we can also then introduce condition with magnesium chloride and see cleavage. So I'll show you what our data looks like, but really we can stop and assess any one of those processes, either in itself or go all the way to the end here and do an actual cleavage assay. And we can repeat that with different guide RNAs. We can repeat that with different amplicons. We can change some of the conditions if needed. But this is basically a CRISPR assay that goes on every day here at CRISPR QC in the lab. Okay, so this is an actual data trace of a cleavage assay. And I like to show it because, we're all scientists and we want to see data. So, but what is really possible to see here is that it's a real-time readout, which means we do get responses that are measurable and quantitative at each of the steps. So I can see how much immobilization has occurred. I can see how much guide RNA has inserted. I can see how much amplicon bind. I can see how much cleavage of the amplicon has occurred. And I like to show this one too, because as Francie pointed out, there are 3 channels on a chip. And this is all 3 channels. This is the 3 channels that are laying on top of each other, which shows you how reproducible and precision-oriented our chips are, or Cardea's chips are. I buy my chips from Cardea. So they make a great chip, and I think some of the questions came around that. The manufacturing process is phenomenal, and the reproducibility of those products that we get from Cardea are absolutely phenomenal. But we get to look at this, and this is what we interrogate. So we can do endpoint analysis at different steps. We can do kinetic analysis, as you might guess, by looking at the fact that the curves have, slopes. So there's a lot of information that can be extracted from a data set that is generated such as this. Okay, so let me now backtrack a little bit, if you will allow me, and I'm going to just talk about kind of major one application in each of those areas I explained in that rectangle box that was kind of between, in silico and cell work. So let's talk about Cas9. I think a lot of people, I would say, that I've been working with most recently with my customers, one of the areas that I kind of open their eyes about, because I'm actually a protein chemist, like Francie said, she's like, I'm not a molecular biologist per se, but I've become one. I'm not a physicist, but I've become one. I'm traditionally trained as a protein chemist. And as everyone knows, proteins are pretty finicky. So one of the things that I've realized in working with customers is that Cas9 kind of gets a hall pass. What do I mean by that? Everyone just assumes Cas9 works like Cas9, and stability, storage, formulation, how long has it been in the freezer? Is it 20, minus 80? Kind of gets a pass. But what we found is when we went and looked at 3 different vendors who produce wild type Cas9. So what we did here is we immobilized just 3 different vendors of Cas9. That was that orange component on the chip surface. And we used our cleavage assay to then assess multiple lots and of each of the vendors' Cas9. So there's a lot of data points here. Each one of these is a chip. And so what you start to see is things like this that may not be intuitively present in your thinking when you're designing your CRISPR assay, which is we have a negative. So what we have here is a negative guide RNA, which shouldn't interact with and cleave an amplicon that would show up in our cleavage assay. We run that control. It's the same one for every assay. It's a very well-designed negative control. And our positive is a very well-studied positive control that we use here at CRISPR QC. And you'll see with vendor 1 that you see all the chip data points are clustered very nicely around 120. The error is, relatively small. And then you would go to maybe vendor 2, who I'm not going to say any of these vendors are making bad Cas9. I don't feel comfortable saying that, but I can tell you that they vary or they differ from one another. So vendor 2 is interesting. They have one lot that's up here around the same value of 120 as, say, these guys, but they've got another cluster from other lots that seem attenuated a little bit lower. They worked because they are still significantly different than the negative, but you have 2 populations of results. And this is one of those results that when you see it, you think, well, from time to time, if I have a CRISPR result and I didn't see what I wanted, is it really because the assay didn't work, or is it possibly because the Cas9 had some variability in it, if that makes sense? Hopefully it does. And then you have vendor 3, and vendor 3 is just different in the sense that, again, the negatives are always around 40. You see that being reproduced, but they have something around 100 and, maybe a little higher than 90 to 120. So they're just a little more spread out, which means we just see a little bit more variability with that cleavage result when we standardize the guide RNA amount and the Cas9 immobilization. So I can tell you that a lot of the customers that I started talking to about an actual CRISPR assay, maybe cleavage, we also now do a bit of work with them studying the Cas9 that they're using, or the Cas12 or the Cas13, because, it's kind of, if you start with something poorly active, you might end up with something less than expected. So we do a lot of this very, very simple analysis for customers now, which is quite rewarding, to be honest with you. And it's a very simple experiment to do. It doesn't take long. It's just the first third of that assay that I showed you. And so we do advertise this and now provide this service to a number of companies. All right, if I take it a step further, and what I mean by that is now I'm going to switch gears on you and take you to guide RNA data, where we actually look at different guide RNAs. And I think this one gets really interesting because, again, I tell you that when you design a guide RNA with the intention of editing, you always get several options. The in silico will say, we would recommend you get these 6 guide RNAs to do your editing. So are they different? That's the question, because you just use them in a cell and then you go, well, they may be the same, maybe they are slightly different, but why? So we try to look at maybe partly as to why there might be differences using different guide RNAs. So this actual customer data, I can't reveal who it is, but it's actual customer data where they sent us 3 different guides here. And the in silico said this should be, all these should work really well. And we look at them by looking at just their interaction after we tethered Cas9 to the surface, and we let the guide RNA come in at 30 nanograms per microliter. We let it bind, we wash everything away, and we look at how much is retained and how strongly bound that is. And what you see is 2 different versions of the same result. The red box is our control negative, meaning that is basically bare Cas9. There's no guide RNA in there. So nothing is really interacting with the surface. And then what you'll see is guide 54, I'm having a hard time, guide 4 is attenuated away from the control, the blue one, the second row. The third one looks significantly stronger in binding, and the last one that's kind of in gray is close to the third one, but it's a little attenuated differently. What we do is we normally normalize this value to 1 in making it the negative control, and we look at the fold difference between these different guide RNAs, and what you'll see is one of these guide RNAs actually is over 12 times stronger in a binder or interactor with Cas9 than say the weakest one or the control, right? So there's a new piece of information. Nobody sees this. We are actually the only company that I'm aware of that can actually take your guide RNAs, as few or as many as you have, and give you some quantitative information about how each of those guide RNAs are interacting with Cas9. That would be an RMP intended to go down into some further work. And so this is now turning out to be a correlative type of association that we're seeing our customers come back to us and say, this is great. We can use this information. We can throw away maybe guide RNAs that we don't want to spend time on. We want to focus on, very strong binders, and there's an optimization and value to that as we go forward. Another guide RNA one, again, customer data, and I have 2 of these just because these are actual customer data that they're now working with us, and this type of information is in fact being utilized. Same process, negative guide RNA. And we have 4 guide RNAs here. And again, 3 of them, if you see 172, 283, 130 on the far right side, all are kind of in the same range, 1.5 to 2 times or 2-fold different. But again, not maybe as impressive as the last one, but 1341 is about 5 to 5.5 times stronger interactor with Cas9 than the other selections possible. Okay, so again, 2 different customers, 2 different data sets, slightly different value ranges, but showing differential display of guide RNA interactions with Cas9. Again, I do not believe there's any technology out there currently that can provide this type of information. All right, the last thing that I'll touch on, which is relatively new, and it came about in part from some of our partners, is a question of when we do form complexes like you saw previously in those slides where we saw differences. Those were actually all done at pH 7.4, just so you know. We use a very standard physiological buffer when we look at the association before and after and determine how much bound. But the question arose, well, not all environments where these editing events take place are at the same pH. There are known cell types transformed, for example, that may have more slightly acidic environments. So what we did is we looked at a couple of things. We looked at different pHs. Here we have 4.5, 6.0, 7.4, and this is the standard assay. And what we did was we did that assay that I just showed previously with a lot more guide RNAs. They're all listed over here. And we ranked them as if the ones that were very tall in their bar chart, we ranked that 1. And then we reassessed all those guide RNAs in a different pH environment. And we wanted to see, does the scoring where 1 would be the best, the higher number would be the worst, does that change? And you start to see like a particularly unique type of guide RNA. It does change. It goes from 1 to 2 to 3, maybe not a big deal there for argument's sake. But you do see other ones where, something could be a 2 at 4.5, pH 4.5, sorry, but actually not a very good interactor at 7. So it just provides additional information to the CRISPR scientist. Do I need to take into account the environment around which my editing may be occurring? And does that need to be taken into account for my guide design? Because I am not aware of right now that any in silico guide design software actually takes into account pH. And I think it's probably because there's not enough of a big data set to do that. But we are creating that information in that data set as we work with our customers in the industry. So I think it's a very interesting type of experiment. I think it's physiologically relevant. I think it's new data points. I think it can be encapsulated into CRISPR checkpoints and CRISPR standards. And I think it'll help CRISPR therapeutic researchers come up with new ways to design their RNPs to do what they want them to do. Okay, the last one is going to be the cleave assay. It is the longest assay. It is embedded with all the information we just talked about. So when I do a cleave assay, I do immobilize Cas9. I do insert a guide RNA. I do apply an amplicon, which does bind, and so I can see that. And then at the very end, I introduce magnesium chloride, and that allows the cleavage to occur. So the one powerful thing about the cleave assay is that you can stop and assess all of that in a single assay. If we're only looking at Cas9 performance, we may not be going as far as an amplicon binding or amplicon cleavage, but with the cleave assay, we do get that. So how do I do that? How do I get rid of this? Can I don't know if I want to leave. That's covering up your slide, right? That's covering up your view. Oh, that's not good. Sorry, guys, my bad. I don't know how to deal with that. That was just to show you that we're at the very end. Monica, is that covered up with what I see, all these different?
I think it looks okay to me. I see 3 bar graphs in the middle of the slide and a little graph with kinetics sort of, up there. Yeah.
I appreciate you telling me that. So I see a bunch of Zoom options, but apparently they're blinded to the audience, which is good. It was only a small graph to let you know that I'm at the very end of that very long assay when I showed you the original larger plot that had the entire data set represented from start to finish. But we really just need to focus on the data set below, right? So this is the cleave assay. So again, Cas9 on the surface. In this case, 3 different guide RNAs, guide RNA 12, 36, 80, which is represented in the graph, and 3 amplicons. Now, I didn't know what they were other than the NC as a control, which means it should be negative all the time. It shouldn't really interact with any of the guide Cas9 RMP complexes that I formed. And so once we did the analysis and we went to the very end of that assay and we looked how much cleavage occurred, was there even cleavage? Did it align with what the customer had? Well, the customer then revealed the table of knowledge to me, if you will. And of course, I already knew these should be negative, but I didn't know how these different amplicons would interact with a particular guide RNA. And if you look at the table, you would hope that 12 would have been negative for the control, of course, plus, plus, minus. And what you see is negative, plus, plus, minus, because we're comparing this normalization of the control to 1 and how much it increased, over the amplicon that was supposed to be specific. So we match there. And if you look at number 36, you would expect negative, of course, plus, minus, plus, and we're negative, plus, minus, plus. Okay, now I got to back up. You could argue that this may not be entirely negative. It is above the negative control, and it's kind of halfway on this one. I can't give you a full explanation of that. We're still working with the customer to try to determine what does that mean. One possible explanation, and we'd have to do more experiments, is that particular amplicon may have some overlap sequence with that guide RNA, which it should not, but it could be leaning a little bit toward is that an off-target event. I don't want to say that it is, not here yet, but that's one of the things we're looking at. It's not quite truly negative, I would say. It's a high enough that, this was a little above, but no, this was noticeable, but it is what it is. And we're still working with that customer to understand that. And if it did, that would be quite interesting. And that would be quite important to know, but I can't stand on that yet. The last graph here, guide number 80 should be negative, positive, negative, negative. And of course, that's exactly what we see. So it's very reassuring, very, very rewarding to essentially go through the entire process, get a little bit of a blinded examination, pull back the curtain and show the customer that in fact we weren't able to get the pattern of positive and negative responses that they in fact expected to get. All right. So at the conclusion then, what I hope that I've been able to show you is that we're able to set up quality control standards upstream of genome editing. And I think the word standards is really important here. Our goal here is to try to empower researchers with a technology where those different checkpoints, the checkpoints of Cas9 activity stability, the checkpoint of guide RNA assignment, assessment, selection, whether or not that RMP complex then has cleavage capability. We want everybody to have the ability to look at those and have a way to assess moving forward so that outcomes are improved, right? It's easy for us to do because we observe everything that happens. That's one of the beauties of the technology is we get to see everything. That's fantastic. Again, I said we have standards now for Cas9, guide RNA, and cleavage activity. And again, the whole idea here is to maintain consistent results and help, individuals make the process of doing something at the end a much more rewarding event. And of course, it saves time and resources. A cleave assay takes me about 4.5 to 5 hours, and we can usually run anywhere from 4 to 8 of those on a single system. I did not mention that the system is fully automated. It's a liquid handling system to which our hardware has been fully integrated, and we have these programmed assays. So there's minimal human error intervention other than the initial preparation of the samples, and we're working on actually having the robot do that as well. So I thank you for your time and listening, and I'd be happy to answer any questions that might also be burning on your mind.